So, as we mentioned in the last video, animals have tissues, these specialized groups of cells that are anatomically and functionally similar and kind of work together. Um, and there are four main types of tissues, ultimately, when in a fully developed animal: epithelial, connective, muscular, and nervous. But these all come from three main germ tissue layers that arise during the embryonic development of an animal. And some animals only have two layers of germ tissue. We call these animals diploblasts, di for two, and diploblasts have endoderm, which is on the inside and forms the gut, and ectoderm, which covers the surface. Triploblastic organisms also have another type of germ tissue, and this is called mesoderm. And mesoderm is in the middle, hence the meso, and that forms, for instance, in us, muscle, bone, and blood, essentially. So sponges don't have any true tissues. They're the exception here, but all other animals have tissues, and they are either diploblastic, having endoderm and ectoderm. This includes cnidarians, like jellyfish and sea anemones. Comb jellies are another group. And then most animals are triploblastic and have mesoderm in addition to endoderm and ectoderm. Another trait that's often used to characterize animals into groups are body cavities. And most triploblastic animals, not all, but most, have a body cavity, which is essentially a tube within a tube. And I mentioned that COEL, seal, refers to cavity. And so organisms with true body cavities are called coeloms. And this is what these look like. So if you can see, this is a cavity right here. And so this is a tube surrounding the tube of your digestive tract. So it's a tube within a tube. And this allows your digestive tract to expand some without messing up your outer skin or your size. It also gives you a fluid-filled sac to squeeze on, because fluid doesn't compress. So that's a hydrostatic skeleton. We'll talk more about that later. But in true coelomates, the coelom is developed in this mesodermal tissue. So in the middle of the mesodermal tissue right there. Some organisms have coeloms, but they're pseudocoeloms, which means fake coelom. Now, functionally, this doesn't mean much, but developmentally, it means that this cavity, this tube that your digestive tube is within, doesn't form within the mesoderm. It forms between the mesoderm and the endoderm, right? So this would be like a nematode. And some organisms, some animals that are triploblastic have endoderm, mesoderm, and ectoderm, don't have any cavity at all. So flatworms, like Platyhelminthes here, don't have any cavity at all. These are called acoelomates, pseudocoelomates, and coelomates or eucoelomates, which means true coelom. And functionally, coelomates and pseudocoelomates have a lot in common, and they're generally placed in the same grade. But this is different than the same clade. So a clade is a group of individuals that include the common ancestor and then all of the descendants. And while it was thought initially that coelomates must all be related and pseudocoelomates must all be related, now with genetic techniques and molecular techniques, it's becoming clear that these are not true clades, right? So they don't include the common ancestor and all of the descendants. Some of these things have evolved multiple times or have been lost across time. Another developmental trait that we use to characterize organisms is whether they are a protostome or a deuterostome in their development. And we described this a little bit in chapter 40 video, but this has to do with whether that initial pore, or the blastopore that forms during gastrulation, is going to form a mouth or an anus. So in protostomes, that blastopore, that first hole, is going to become the mouth, and protostome literally means first mouth. In deuterostomes, which means second mouth, another pore will open up and become the mouth. That first pore will be the anus. So the mouth comes from the second hole, hence second mouth, but we develop anus first. I say we because chordates, including human beings, are deuterostome. And as you can see, the end result doesn't look much different, but the way you get there is different. And this does turn out to be relatively instructive in terms of clade. And it turns out this protostome development or deuterostome development are associated with other developmental traits as well. So protostomes tend to undergo spiral cleavage, whereas deuterostomes undergo radial cleavage. So in radial cleavage, you can see these lines where you have cleavage forming between the cells, kind of in this, 4, 8, 16, 32 cell stages. You could draw a line down here and you're going in the same direction. Whereas for spiral, you draw a line down between the two cells on top, and you have to scooch over to see where the line is between the two cells in the bottom, right? It's almost like you turned a Rubik's Cube, right, into a spiral, hence spiral cleavage. So deuterostomes have radial cleavage, protostomes have spiral cleavage. Deuterostomes also have indeterminate cleavage, whereas protostomes have determinate cleavage. Determinate cleavage means that almost immediately, right after that first cleavage from 1 to 2 cells and then 2 to 4 cells, those cells are now different, and they are differentiated enough that they have to become certain ends of the body. And if you were to break those cells apart, then that organism is dead, right? You can't keep going. Whereas deuterostomes have indeterminate cleavage, which means for the earliest stages, so 2, 4, 8, when those initial cells are there, if they break apart, they're still unspecialized enough that those 2 new clumps of cells can each continue developing, and they'll become 2 organisms. And that's actually how identical twins come about, right? So we are deuterostomes. We have indeterminate cleavage. And in fraternal twins, mom just happens to ovulate 2 eggs in a month, and 2 of dad's sperm, make their way up there, and 2 different sperm fertilize 2 different eggs. They just happen to be there at the same time in mom's uterus. But for identical twins, which are sometimes called monozygotic twins, a single sperm and a single egg came together, and during cleavage, they broke apart. And so if you're identical twins, you are genetically identical to your sibling, and you came from the same zygote, right? The same sperm and egg. Additionally, protostomes, the mesoderm that you find in protostomes forms out of these solid masses that are made of mesoderm between the endoderm and the ectoderm, whereas in deuterostomes, the mesoderm forms from these little buds on the end of the endoderm and then, kind of comes out. But anyway, all of these are associated with either protostomes, things like spiral and determinate cleavage and first mouth from the blastopore, or deuterostomes, you have radial and indeterminate cleavage, and the blastopore becomes the anus, and you form a second mouth later. There are about 36 phyla of animals, and we are not going to learn every single one, but we are going to move through a lot of the major ones that have sort of the most diversity when we get to chapter 33 and chapter 34. But how you combine these into a phylogeny, how you retrace the evolutionary history of animals, is difficult, right? We weren't there to watch it, and we have evidence that is robust in some areas and limited in other areas. And so every time a scientist puts forth a phylogeny, it's a hypothesis of what the history of the evolution of these organisms looked like. And so there are still current debates in animal systematics, which is the study of how you group these organisms into a phylogeny, about how they should be placed. Now, one hypothesis is a little bit more old school because it's based mainly on morphological and developmental characteristics, and these are things that we've had the technology to look at for a long time, and it looks something like this. And we have some common ancestor of all metazoans, all animals, right? Zoa means animal. And then the first to branch off were the sponges, who are the exceptions to so many of these rules that we've talked about. And it makes a lot of sense that they're on their own. And then the common ancestor of all of the rest of the animals was right here. And we put this group these animals into a group called Eumetazoa, which means true animals, right? Sorry, sponges. These are the true animals. And that includes all organisms with tissues and symmetry, right? Because sponges don't have either of those. And our first organisms to branch off here are Cnidaria and Ctenophora, which are like jellyfish, and comb jellies, which are radially symmetrical, diploblastic, look a lot like comb jellies. And then these are radially symmetrical organisms. Then we have a group called Bilateria, where we had a bilaterally symmetrical common ancestor, and we separate this into two main groups, Deuterostomia and Protostomia, and these are all major phyla of organisms that we're going to discuss. Now that we have molecular techniques, we can look at the genes, the A's and T's and C's and G's in the DNA of these organisms. We can look at the protein structure, the amino acid sequences, things like that. You get a slightly different hypothesis for how animals evolve. And we still have, metazoa, which is all animals, right? Some common ancestor to all animals, with sponges branching off first. And then you have eumetazoa with Ctenophora and Cnidaria, which are radially symmetrical. And you still get then the group Bilateria with a single common ancestor. But instead of just having Deuterostomia and Protostomia, we have a smaller group of deuterostomes that includes Echinodermata, like sea stars, and Chordata, us. And then you have two weird groups, right? Lophotrochozoa and Ecdysozoa. And this mixes up how we used to characterize these phyla, right? We used to think that, for instance, Annelida and Arthropoda must be very closely related because they have segments, right? And we thought segments must have evolved in a common ancestor. Well, the molecular phylogeny says no, segments evolved two different times, once in this group and once in this group. And Arthropoda, instead of being with Annelida, should be with Nematoda because they both have cuticles on the outside that they molt. And so we call this group Ecdysozoa, ecdysis meaning molting. And we will discuss some other hypotheses, right? Some other areas of disagreement. There are still, there's new data being put out there and published as we get better and better at molecular technology. But there are points of agreement generally. Most phylogenies agree that sponges are the basal animals. Eumetazoa, meaning all animals, are a true clade, which means it's a group that includes the common ancestor and all of its offshoots, right, descendants in one group, one clade. Most animal phyla belong to the clade Bilateria, bilaterally symmetrical organisms, which also is a clade, meaning it's a solid group with a good evolutionary foundation with one common ancestor and all of the descendants. And that the chordates belong to a group called Deuterostomia with a sister group of deuterostomes that includes echinoderms. Most of this is agreed upon. Within Bilateria, all right, our molecular phylogeny creates this new group called Ecdysozoans, right, which pairs arthropods with nematodes again. So instead of being paired based on their segments, they're paired based on ecdysis, this process of molting. And we have this group Lophotrochozoa, which crams a bunch of different groups together that we thought were separate, looking just at morphological characteristics, but molecularly it looks like they all belong together. By the way, when I say ecdysis or molting, this is what I mean, right? You leave behind your old skin, essentially, and you've probably seen a lot of these molts sitting around. You certainly will next summer when we have the cicadas come back. And while a lot of the group Lophotrochozoa, which is a group of different animal phyla, may seem like a mess and they're only together because of the molecular systematics, there are a lot of them which are together because of these structures, these lophophores. And so they're lophophorates, right? And there is another group that are there because of this trochophora larva that they all share in common. And so these two traits, this larva and this lophophore feeding structure, are why we call this Lophotrochozoa. And as homework, I would like all of you to say Lophotrochozoa 5 times very quickly.